The key cells of the adaptive immune response are the lymphocytes, the B and T cells. And there are two types of T cells: T helper cells, which express CD4 on their surface, and cytotoxic T cells, which express CD8 on their surface. Helper T cells primarily support other immune cells, whereas cytotoxic T cells kill cells that are infected with a pathogen or are cancerous. Cell-mediated immunity refers to the part of the immune response that's based on cellular interactions and cannot be transferred through serum from one person to another. That makes sense since CD4 helper T cells interact with other immune cells to stimulate them. Now, when a T cell is initially formed, it's considered naive. This naive T cell is a bit like a student in school that isn't ready to choose a career path. Later, when that T cell encounters an antigen, it gets activated, or primed, and turns into an effector T cell. This process of priming requires two signals. The first signal is the antigen itself, which is usually presented on an MHC molecule on the surface of an antigen-presenting cell like a macrophage or dendritic cell. This antigen has to bind perfectly to the T cell receptor. The second signal is called co-stimulation, and that's when a ligand called CD28 on the surface of a T cell binds to a ligand called B7 on the antigen-presenting cell. This region, which includes the T cell receptor, which binds to the MHC antigen, and CD4 and CD28, which bind with B7, is called the immune synapse. Once the T cell receives both of these signals, a number of changes occur within the cell that transforms the naive T cell into an activated T cell. An activated T cell is one that's ready to become an effector T cell, and the developmental path it chooses often depends on the cytokines in the environment. Going back to the student analogy, an activated student is one that's gotten the signals it needs to graduate from college and is finally prepared to choose a career path. Now, within the broad category of T helper cells, there are subcategories like T helper type 1, or Th1, T helper type 2, or Th2, T helper type 17, or Th17, and T follicular helper cells, or TFH. There's also a class of T cells called T regulatory cells, or Tregs, but these aren't considered T helper cells because they're mostly involved in contraction of the immune response. Within the immune synapse, antigen-presenting cells, or APCs, send out different combinations of cytokines depending on the type of pathogen. These cytokines help tailor the immune response to the specific pathogen. The activated T helper cell also begins making lots of cytokine IL-2, and it upregulates its IL-2 alpha receptor. The IL-2 receptor has three protein components: alpha, beta, and gamma. A naive T cell only expresses the beta and gamma components of the IL-2 receptor, but these have low affinity for IL-2, in contrast to the alpha component, which has a high affinity for IL-2. Using just the beta and gamma components is kind of like trying to eat an apple with only your lips and tongue, and upregulating the alpha component is like using your teeth to take a big bite out of that juicy apple. Much more effective and satisfying. As a result, active T cells bind to IL-2 that they make, a form of autocrine stimulation, that is to say, a cell stimulating itself. In response to the IL-2, the activated T cell starts to rapidly undergo cell division, a process called clonal expansion. Now, let's go through the types of T helper cells, the career paths, one at a time. Macrophages and dendritic cells infected with viruses and intracellular bacteria produce IL-12, and natural killer cells produce interferon gamma. In addition, any virally infected cell will produce interferon alpha and interferon beta. When these cytokines are found in the same area as a T cell that's becoming activated, they bind to the T cell and cause it to make transcription factors like STAT1, STAT4, and TBET. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th1 cell. A Th1 cell then secretes interferon gamma, which reinforces the process. Interferon gamma also boosts the macrophage's ability to kill ingested pathogens by increasing their production of reactive oxygen species, nitric oxide, and lysosomal proteases. Th1 cells also express CD40 ligand on their cell surface, and that binds to the CD40 receptor on macrophages, which also stimulates them. Th1 cells also produce IL-12, which makes natural killer cells more effective. And they produce IL-2, which makes natural killer cells, CD8 positive T cells, and B cells proliferate. So in short, Th1 cells stimulate themselves, and they stimulate a number of other immune cells as well. When Th1 responses are not carefully controlled, this can sometimes lead to autoimmune reactions. Next up are the Th2 cells. When there's a parasitic worm that's too large to get phagocytosed, it gets destroyed by eosinophils, basophils, and mast cells, which dump their toxic granules full of proteases and hydrolases. This kills the parasite, and its antigens get picked up by scavenging macrophages and dendritic cells. These antigen-presenting cells produce IL-4, IL-5, and IL-10 within the immune synapse with a T cell. When these cytokines are released within immune synapse, they bind to the T cell and cause it to make transcription factors like STAT6 and GATA3. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th2 cell. Once it's become a Th2 cell, it produces cytokines like IL-4, IL-5, IL-10, and IL-13, which stimulate B cells to make IgE, which can coat the worms. In addition, Th2 cells stimulate mast cells, basophils, and eosinophils to bind the Fc region of the IgE and degranulate so that they can kill the parasite. Unfortunately, Th2 responses can sometimes get directed at non-pathogenic antigens, like peanuts or tree pollen, and this results in allergies, atopic dermatitis, and asthma. IL-13 specifically stimulates mucus secretion and intestinal peristalsis, which can help remove parasites from the intestines. Finally, the IL-4 and IL-5 help to stimulate the Th2 cells themselves, which reinforces the process. Now let's move on to the Th17 cells. Extracellular bacteria and fungi are often picked up by dendritic cells or macrophages, which recognize bacterial peptidoglycans, lipopeptides, or fungal glycans. These antigen-presenting cells produce IL-1, IL-6, IL-23, and TGF-beta within the immune synapse with a T cell. When these cytokines are released within an immune synapse, they bind to the T cell and cause it to make transcription factors like ROR-gamma-T and STAT3. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a Th17 cell. Once it's become a Th17 cell, it produces cytokines like IL-17, which is where it gets its name. The main function of IL-17 is to recruit neutrophils, which engulf these extracellular pathogens and destroy them. When Th17 responses aren't carefully controlled, they can lead to autoimmune disease and asthma. Finally, there are T follicular helper cells, which are a special group of T cells that live in the follicles of lymph nodes. Their primary role is to help generate memory B cells. Occasionally, antigen-presenting cells produce IL-6, IL-21, and IL-27 within the immune synapse with a T cell. When these cytokines are released within an immune synapse, they bind to the T cell and cause it to make transcription factors like BCL6 and CMAF. These transcription factors cause a number of genes to get expressed within the T cell, transforming it into a TFH cell. Once it's become a TFH cell, it expresses CD40 ligand on its cell surface, and that binds to the CD40 receptor on B cells and makes them express cytokine receptors. The TFH cells also produce cytokines like IL-21 and interferon gamma, which stimulate B cells to make IgG antibodies and become memory B cells. Alright, as a quick recap. Immune responses are determined by the cytokines the T cell is exposed to within the immune synapse at the time of activation. Th1 cells fight intracellular infections. Th2 cells fight parasites. Th17 cells fight fungal and bacterial infections, and T follicular helper cells help establish memory B cells.